So I should go back more than 50 years when a Hungarian Nobel laureate, Albert Szent-Györgyi, who discovered the vitamin C, visited Hungary. And in his interview, he said, if you want to solve the cancer problem, you have to go sub-molecular level. I was very young, teenager at that time. And Albert Szent-Györgyi, he was a very clever Hungarian scientist because his way of thinking was that how is it possible that we can and the cells can respond so fast for all the challenges. Meantime, the proteins are so rigid, they are so big. So he raised the question whether is it possible that the life is organized by that molecules, big proteins, which the synthesizing the proteins takes long time, the induction of the genes takes long time. So that word submolecular, that was the very first point which was very important in that long journey. So I kept it in my mind that submolecular, and Albert suggested that particle should be that negatively charged electron. And one day, that was in 1976, May 21st, I thought, okay, maybe the proton should be that submolecular particle, the positively charged hydrogen ion. So I make a note and I wrote down that the hydrogen ion responsible for the cancer. And that was in 1976. And then I went to the university, and 4 years later at the university, the other idea came to my mind that there is a heavy hydrogen, it is called deuterium. And at that time, that was in 1980, I was sure that somehow the deuterium and the hydrogen together regulate the cell growth and responsible for the tumor development. I graduated in 1982, then I continued my work, that was a DNA work, I isolated genes and identified genes. I had a fellowship holder for the Hungarian Academy of Sciences. I won my PhD. I worked in Germany. I went to the United States. When I came back from the United States, I started to look for a job which is related to the cancer because that was my original plan to do research on the cancer field. So in 1990, I started to work in the Hungarian National Institute of Oncology. And my question was to investigate whether this deuterium has any role in living organisms. The people should know that the deuterium was discovered almost 100 years ago. And it was clear that due to the 100% mass difference between the hydrogen and deuterium, that these two hydrogens behave differently in chemical reaction. It is called isotopic effect. And for 60 years, the heavy water was tested in different biological systems. And it was clear that it's for a living organism, it's not the same having a deuterium or hydrogen, because you can kill a dog with a heavy water over 35%. So it was clear that even both is hydrogen, in the chemical aspects, we can talk about two different elements. And the other disturbing thing was, I guess, in the last 100 years, that the ratio between the hydrogen and deuterium is about 6,600 to 1. So if we take 1 million hydrogen atom, there will be only 150 deuterium atom out of 1 million hydrogen atoms. So this is the reason we say that the deuterium level is about 150 ppm parts per million. And I suppose that the scientists, because that ratio, 6,600 to 1, believe that, okay, let's ignore the deuterium. It is so rare. Why should we think about deuterium? And that was a question what I addressed. How can we ignore the deuterium? If we express the deuterium level in millimole, it turns out that deuterium level in our body is 6 times higher than calcium and 10 times higher than magnesium. So this is the question I addressed. What will happen if we reduce the deuterium level, and what kind of consequence we can detect in the biological system? So in the very first paper was published in 1993, and in that paper we proved that the lower D level reduced the cell growth in vitro, and when we replaced the drinking water of the mice, which had been transplanted with breast cancer, we could save the life of the mice, which is clear indication that first, the deuterium-hydrogen ratio is critical and has a big impact on the cell growth. The second, the deuterium depletion has an anti-cancer effect, because in these mice, the cancer disappeared when the mice started consuming DW. So when we received that type of results, I quit at the institute and we established the company, the HYD, and I promised my partners that we will register the best cancer drugs of the world. That was more than 30 years ago, and we are still working on that to convince the world and to get the approval and to take part in the depletion of the oncotherapy.
Right.
And one of the questions always, whether we know the mechanism. And we say that, yes, we already know the mechanism because we have invested so many time and fun to figure out what's going on. So everybody every day synthesize about 0.4 liter deuterium depleted metabolic water. So when we are eating the food and we burning it, carbon dioxide and metabolic water, that metabolic water is deuterium depleted. And how depleted, it depends on what type of food we eat. The experiment we done was very simple. We went to the supermarket, we bought different kind of food, and then we measured the deuterium level of the dry components of the food. And it turned out that from the carbohydrate, the cell synthesized metabolic water with 150 ppm, so like normal deconcentration. But when we burned the fat oil, that was 118 ppm or 109 ppm. So that means the more fat we eat, the lower will be the deconcentration of the metabolic water. Yeah. And why is it important? Because the brake system in the cells is in the mitochondria, and the mitochondria producing the deuterium depleted metabolic water. And the brake is very important because when a growth hormone binds to the membrane and trigger the cell growth, that will stimulate the so-called sodium-hydrogen exchanger, which means that the cells pick up one sodium and transport one hydrogen. And in our very first paper, we recommended that when the sodium-hydrogen transport is activated, that will be separately transfer only to hydrogen, which means the activation of the sodium-hydrogen antiport will cause an increase in the DH ratio. Yeah. And we said this is the signal for the cell, which are when the DH ratio within the cell reaches threshold, that will turn on hundreds of genes, which expression is essential to manage the G1 phase to the S phase, doubling the DNA, and then close the cell cycle. So in a healthy cells, the membrane transport can increase the DH ratio, but in a healthy cells, the mitochondria produce deuterium depleted water, so they are imbalanced. Yeah. And this is another story which is well known for almost 100 years, that the only common feature in each cancer is the damaged mitochondria. So in cancer cells, the mitochondria is not able to produce deuterium depleted metabolic water. So this is the reason when there is the activation of the growth hormone, stimulate the sodium hydrogen antiport, that much easily can lift up the DH ratio to the threshold, and then the cell will divide and will multiply. So that's it. Right. And so when, and the very important message, when we modify the DH ratio, that will simultaneously modify all the enzymes, because will be a new equilibrium for the DH ratio. So when the DH ratio is increasing, then in each molecules, in the proteins and all the molecules which has a hydrogen in an exchangeable position, then more deuterium will bind in that protein and other molecules because we increase the DH ratio. So that sends a message to all the molecules. And this is how the cells can be organized, because it is so complex. But even the complexity, it is well organized and can manage all the challenges. So this is a basic mechanism.